Hello students. So today we will discuss about chemistry of amino acids and proteins. The contents of today's class will be amino acids. What are amino acids we will discuss and their classification, definition of protein, structural organization of protein and the tests for proteins and amino acids and the questions relevant to this topic. Now, what are amino acids? Amino acids are the building blocks of the proteins. Proteins are composed of a number of amino acids linked by the peptide bonds. Although about 300 amino acids occur in nature, only 20 of them are seen in human body. Unlike the carbohydrates, all amino acids in human physiology are L-amino acids. We have seen in carbohydrates that most of the carbohydrates which are present in our body are in the D form, whereas for amino acid, the L-enantiomer is the most prevalent form. Structurally, all the amino acids are alpha amino acids except the proline and the hydroxyproline, which are amino acids. Now, the amino acids are classified on the basis of structure, the character of the side chain, their metabolic fate, and nutritional requirement. Now, let us discuss classification of amino acids on the basis of structure. On the basis of structure, amino acids are alpha amino acids like glycine, alanine, valine, leucine, isoleucine. Aromatic amino acids, these are mainly the phenylalanine and the tyrosine. And heterocyclic amino acids like the tryptophan and histidine. And amino acid, as I have already said, it is proline and the hydroxyproline, which is derived from the hydroxylation of the prolyl residue. Now, derived amino acids are amino acids which are derived from the post-translational modifications of the amino acids. These are mainly hydroxyproline and hydroxylysine, which are abundant in the fibrous protein collagen, and gamma aminobutyric acid and beta alanine. These are also derived amino acid and not found in protein. Non-protein amino acids, what does they mean? They mean that they are amino acids, but they are not found in protein, neither they are translated from any of the RNAs, yet they are amino acids. These are mainly ornithine, citrulline and homocysteine. Classification of amino acids on the basis of their side chains. Amino acids have non-polar side chains. These include alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. Amino acids having uncharged or non-ionic polar side chain. These are mainly cysteine, threonine, serine, and tyrosine. Among them, serine and tyrosine and threonine, these are hydroxyl group containing amino acids. Amino acids are also having some charged or ionic polar side chain. These are acidic amino acids which are aspartate and glutamate, and basic amino acids which are mainly histidine, arginine and lysine. So three basic amino acids are present in our body: histidine, arginine and lysine. Classification of amino acids depending on metabolic fate. These are of three categories: ketogenic, glucogenic and both ketogenic and glucogenic. There are two amino acids which are purely ketogenic. These are lysine and leucine. Both glucogenic and ketogenic are isoleucine, phenylalanine, tyrosine, tryptophan, and these are either partially ketogenic and partially they are glucogenic, and purely glucogenic are the remaining 14 amino acids, as they enter only the glucogenic amino acid path. Now, classification of amino acids on nutritional requirement. These are essential amino acids. What do we mean by essential amino acids? Those amino acids whose carbon skeleton cannot be synthesized by human beings, and so preformed amino acids are to be taken in food for normal growth and optimum physiological function. These are called essential amino acids. Previously, the histidine and arginine were thought to be in the category of essential amino acids, but later it was found that this histidine and arginine are specially required for the children in the growing age group and in the pregnant and lactating mothers. So the histidine and arginine are thought to be as semi-essential amino acids. Essential amino acids are mainly these 8 amino acids: leucine, isoleucine, threonine, glycine, methionine, phenylalanine, tryptophan, and valine. Rest of the amino acids can be translated from the RNA in the body, and they are categorized as the non-essential amino acid. That means they need not to be taken in the diet or food. The human cell can synthesize them from the. Now, the proteins. Proteins are the polymers of the amino acids that are linked mainly by the peptide bonds. This peptide bond has a special character. This peptide bond is rigid in nature. All the functional groups involved in the peptide are coplanar, that is, they lie in the same plane, and normally the peptide bond is trans in character and it has a partial double bond like character. So there are the 4 categories or important features of the peptide bond. First, it is rigid. Second, it is coplanar. Third, it has a partial double bond character. Fourth, it is trans in category. However, some of the amino acids also form cis peptide bond, like the prolyl and the hydroxyprolyl residues. If they are present in some protein, they favor cis peptide bond formation. Proteins have different level of structural organizations. These are mainly primary structure, secondary structure, tertiary structure, and quaternary structure. The number and sequence of amino acid mainly determines the primary structure. Each amino acid in a primary structure is called a residue. The primary structure is mainly formed by the peptide bond. That is, this peptide bond forms the backbone of the primary structure. The peptide bond has a partial double bond character and most of them are trans in nature, as I have already mentioned. And other 2 important features of peptide bond is it is rigid and coplanar. The intra-chain disulfide bonds are also there in the primary structure. That means if we have any cysteine residue in the primary structure, the cysteine residue between the chains may form disulfide linkages among themselves. Secondary structure is the steric relationship of amino acids close to each other. The hydrogen bond forms an important part of secondary structure organization. There are 2 types of secondary structure. One is alpha helices and second one is beta pleated sheets. Alpha helices, if we look, this alpha helices are mainly present in the heme containing protein like myoglobin, and beta pleated sheets are mainly present in the silk fibroin. Now, the tertiary structure. The tertiary structure denotes the overall arrangement and interrelationship of the various regions or domains of a single polypeptide chain. That means other than the peptide bond and hydrogen bond, the van der Waals interactions, electrostatic interactions also come in play in the organization of tertiary structure of proteins. Quaternary structure. Quaternary structure results when proteins consist of two or more polypeptide chains held together by non-covalent forces, like in cases of hemoglobin molecule contain more than one polypeptide chain. It consists of two alpha chains and two beta chains if we consider the adult hemoglobin. So these are the four levels of structural organizations of protein. Primary, mainly the peptide bond and the intra-chain sulfide linkage. Then comes the secondary, where we have the two categories, alpha helices and the beta pleated sheets, then the tertiary structure, and last is the quaternary structures. If you can see the flow chart here, primary structure, which is determined by the number and sequence of amino acid, each amino acid being termed as a residue. Secondary structure, when the amino acids are linked by the hydrogen bonds, and tertiary structures are formed when alpha helices and beta sheets are held together by weak interactions. What are those interactions? Van der Waals interactions. And quaternary structure consists of more than one polypeptide. Now these are the tests for protein. The tests for protein are categorized in three classes: coagulation tests, precipitation tests, and the color reaction. So in the coagulation test, the first test is the heat and acetic acid test, or the heat coagulation test. Two-thirds of the test tube is filled with the sample solution, and upper part of the sample or test tube is heated. When we heat the upper part of the test tube, a white coagulum appears. As you can see in this figure, there is a dense white coagulum in the upper part. Appearance of white coagulum means protein, phosphate and carbonate is present. Now we will add few drops of dilute acetic acid. If the coagulum intensifies or persists, that means protein is present. If it disappears, that means phosphates and carbonate is present. As soon as you add dilute acetic acid, if it is carbonate, the carbon dioxide will effervesce out, and if it is phosphate, it will dissolve. But in case of protein, whenever we add the dilute HCl, we drop the pH close to the isoelectric pH, and as a result, the coagulum intensifies. What is coagulation? Coagulation is a process by which we do the denaturation of protein. That means the primary structure is kept intact, but the secondary and the tertiary structure is uh, destroyed. This is Heller's nitric acid test. It is also a type of coagulation test. 2 ml of concentrated nitric acid is taken in a test tube, and then the sample is added drop by the side of the test tube. A white ring appears at the junction of the two liquids, which infers that protein is present in the given sample. So this is the figure of Heller's nitric acid test, or Heller's nitric acid. Next is the precipitation reaction. Precipitation reaction means it consists of two types of tests, that is lead acetate test and H-back test. Lead acetate test means 2 ml of test solution is taken in a test tube. 1 ml of 5% lead acetate and 0.5 ml of 40% sodium hydroxide is added to it. The mixture is boiled for 1 to 2 minutes and cooled under tap water. A black precipitate appears. That means sulfur containing amino acid are present. We know our there are two sulfur containing amino acids. One is cysteine, one is methionine. Cysteine gives the test positive, but methionine does not give the lead and acetate test positive. Why? Because cysteine has the sulfur hydryl group, and this sulfur hydryl group reacts with lead acetate and black precipitate of lead sulfide is formed. Whereas methionine does not have the lead sulfur hydryl group, the terminal sulfur group is occupied with the methyl group. So it won't react and we don't get a black precipitate. Ashbach's test: 2 ml of test solution is taken in a test tube and 2 ml of Ashbach's reagent is added. We get a yellow precipitate and the inference is protein is present. If you see the composition of Ashbach's reagent, it contains picric acid and citric acid. So, we get the uh, precipitate of protein. Why? Because the citric acid drops the pH of the protein solution below the isoelectric pH. As a result, the amino acids or the protein which is present in the solution becomes protonated, and that reacts with the picrate anion to form the yellow precipitate. The citric acid drops the pH of the solution below the isoelectric pH. What is isoelectric pH? It is that pH where the number of positive ions or the negative ions in a solution is equal. That means there is no net electrophoretic mobility. So as soon as the pH drops below the isoelectric pH, the picrate anion will react with the protonated amino acid and you will get a yellow precipitate. Next one is the color reactions. The color reaction is the biuret test and mean hydrogen test. About 5 ml of unknown sample is taken in a test tube. 1 to 2 drops of 1% copper sulfate and 1 ml of 10% sodium hydroxide is added and shaken thoroughly. Violet color is produced. As you can see in this figure, protein is present. This is due to a coordinate complex formed between the cupric anion of the copper sulfate and the amino group of the peptide bond. Mean hydrogen test. To 1 ml of test solution, few drops of freshly prepared mean hydrogen 0.2% is added and boiled and a purple color appears. This is for the alpha amino acids. This mean hydrogen test is also known as the general test for amino acids. Now what are the questions? Why upper part of the test tube is heated in heat and acetic acid test? Because the lower part acts as the control. Upper part, if we see the coagulum appears, the lower part will serve as a control. Why do protein intensify and rest disappears in adding acetic acid? Because when we add the dilute acetic acid, the pH drops below the isoelectric pH and the coagulum intensifies. If there is phosphate, it will dissolve in the acidic solution. If there is carbonate salt, the carbonate salt will react with the dilute HCl and we will see carbon dioxide coming out as an effervescence. Do all the sulfur containing amino acids give lead acetate test positive? No, it is only the cysteine which give the lead acetate test positive. Methionine does not give lead acetate test positive because methionine does not have free sulfur hydryl group. What is the principle of Ashbach's test? I have already discussed. It contains two acids, picric acid and citric acid. The citric acid drops the pH of the test sample solution below the isoelectric pH, for which the amino acids or the proteins become protonated and it reacts with the picrate anion to form the yellow precipitate. Why biuret test is called biuret? Because this test was first given by heating two molecules of urea in alkaline media at 180 degrees centigrade. It forms a biuret complex, and that gave the test first positive. So after that name, this test is known as biuret test. What is the principle of the ninhydrin test? All alpha amino acids reacts with ninhydrin to form a complex which gives the violet color for the alpha amino acid, and this is known as the Ruhmann's purple. Do all amino acids give purple color? No. Amino acids, what are those? Proline and hydroxyproline do not react with ninhydrin to give purple color. Instead, they give yellow color. What is the normal amount of protein excreted per day? In human, it is the traceable amount of protein excreted per day. It is less than 150 milligram per day excreted in an adult healthy human. What is Stam-Hospel protein? Normally, the protein that is excreted in urine, it consists of Stam-Hospel protein, few enzymes, glycoproteins, apolipoproteins. This Stam-Hospel protein is a uromucoid which is secreted by the distal convoluted tubules of the kidney. What is microalbumin? In human, the albumin in urine is excreted in traceable amount. Due to this minute quantity of albumin that is excreted, it is known as microalbumin. Normally, microalbumin is excreted less than 30 milligram per day in an adult healthy human being. That due to the minute amount, it is called microalbumin. It is not because the size is less. It is the less amount of albumin that is excreted per day. So it is called microalbumin. What is Bence-Jones protein? These Bence-Jones proteins are nothing but light chain proteins, light chain immunoglobulins which are excreted in multiple myeloma. I have already discussed when I was discussing the antigen antibody interactions that immunoglobulin consists of any of the 5 varieties of the heavy chain and the light chains either of the kappa or the lambda variety. This Bence-Jones protein have a peculiar property of denaturation and renaturation. If you go for heat and coagulation test, if you heat it at 40 to 60 degrees centigrade, the coagulum appears. Now, if you further heat it, the coagulum disappears, and again on cooling, the coagulum reappears. So, this peculiar and particular property specific to the light chains which are excreted in excess amount in multiple myeloma, these are known as Bence-Jones protein. Thank you. If you have any questions, you can mail me regarding this topic. Have a nice day.